Hi there, and welcome to Lecture 5 in Quantitative Methods for Biologists. Today we're talking about the principles of hypothesis testing. Hypothesis testing lets us ask how unusual it is to get a specific set of data and how different the data is from a null hypothesis. The null hypothesis is very much the basis for statistical testing. Essentially, the null hypothesis is the kind of the boring statement. It's the statement of no difference, pattern, or trend, and it's sometimes referred to as HO. Hypotheses come in pairs, and the other side of the pair is the alternate hypothesis. And this is where we state the interesting part of the investigation. This is generally where we state the difference, pattern, or trend that we're looking for in our experiment. And this is referred to as HA. What we do in hypothesis testing is we run a little test, and that test allows us to decide if we have sufficient evidence to decide that the HO, the null hypothesis, is untrue. And if it is untrue, we're going to reject it. One thing we have to think about, though, is in hypothesis testing, we're working with the sample, the little group of guys over here, but we're really trying to find out what's going on in the population. The thing we have to keep in mind is that any sample is going to differ slightly from the population. And so long as those methods of grabbing the sample are random, we're actually going to have difference from one sample to another, aren't we? So we always have to be aware that we have to keep in mind this difference between samples and populations. And you'll see that the different statistical tests we use have got different ways of incorporating a consideration of that issue. Okay, 3 basic rules when we're working with hypothesis testing. First of all, the null and the alt must be mutually exclusive. Now that's something we've covered in class, isn't it? So that should be pretty basic. Secondly, the null hypothesis is the simple statement, whereas the alt is the statement of greatest interest. That's where our claim usually is. And thirdly, a good null hypothesis is going to be interesting if we prove it wrong and we can reject it. And at that point, we'll have an interesting scientific finding. Okay, so how are we going to do this? Well, if you can imagine that the outcome of our statistical test is a number called a test statistic. And what we're going to do is have in mind this distribution of that test statistic if null is true. And then what we'll be doing when we test different sets of data is we'll be matching our test statistic with the null distribution for that test statistic, the distribution of that test statistic that you would get if the null was true. Essentially, if you imagine the area under that curve is a whole unit equivalent to 1. So we can use the position where the test statistic falls to give us an idea of the probability of getting that test statistic if the null hypothesis is true. Okay, let's use an example, and this will be much easier to explain. In this example, we are going to be discussing the color of shirts that people were wearing in wrestling bouts. And we're asking the question, does a red shirt help you win when wrestling? There is an idea in nature that red is a sign of aggression. Some animals have the red color and have the aggression, and some animals just have the red color. They're kind of faking it out. So it's interesting to see if this plays out in things like matches such as wrestling, taekwondo, and boxing. In the example we're looking at, there were 16 of 20 rounds in which a red-shirted athlete beat a blue-shirted athlete in this type of sport. And if you know anything about wrestling, which to be quite honest I don't, but in this example it explains that the color is randomly assigned to the wrestlers. So there's always one red and always one blue. It's not a team affiliation thing. And those colors are randomly assigned at the start of the bout. And so the researchers here were asking the question if this unusually high number of red-shirted winners was actually something to do with the color of the shirt they were getting assigned. Okay. So, stating the hypothesis here, we would be laying out a framework of how we expect this test to go. Our HO would be that red and blue shirted athletes are equally likely to win. so the proportion would be 0.5. And IHA would be that red and blue shirted athletes are not equally likely to win, and there was a difference in the proportion of outcomes from 0.5. Okay. Now 16 out of 20 actually did win, and that's a proportion of 0.8. And the difference between this and what we expected is 0.3, isn't it? Because we expected 0.5 and we got 0.8, so our difference is 0.3. So the question we have to ask is if this discrepancy is caused by chance alone. To do that, what we'll do is we'll use the null distribution for the test statistic as our model, and we'll see where our actual statistic that we derive, in this case it's just the probability, falls along that scale. Okay. So that's a little confusing, isn't it? So maybe a picture will help. As you can see here, we've got a frequency histogram that displays the frequency of different scores that you would get with 2 dice, something we've discussed before. Let's imagine here, though, that the frequencies are expressed in proportions, and the total amount of red that you can see adds up to 1, and the columns in each individual section reflect the proportion of the curve that's in that section, and they reflect the probability of actually getting that score with the dice. So basically you can see that 7 has a really high column because that's the most frequently occurring score. And these rarely occurring scores, such as 12 and 2, have much less, don't they? So their column, the size of their column as a proportion of the whole, represents the probability of getting that score. So the null distribution of the sample proportion lets us calculate the probability for our outcome. So this is the null distribution for the sample that we took. These are all the wrestling bouts. Yeah, so there's the 20 wrestling bouts. Okay. And the number of red winners was 16, wasn't it? Now we're going to do a two-tailed distribution, so we're going to be considering whether there's any difference. So we're going to consider this portion and this portion of the curve, and we're going to be asking the question if the p-value we calculate represents a very unusual likelihood on this particular chart, so or against this particular distribution. Okay, so the p-value is calculated as twice the column size for 16, 7, 18, and 19, 20 plus this one over here, and that's where the twice comes from. I'm not doing very well here, am I? Got my things right. Okay, let's stop right there. Let's see if we can pick it up. So what we can do here is we can look at the distribution we would get for the number of red winners, assuming that the null hypothesis is true and there's no difference in red and blue winners. So this histogram that you see right here actually is showing you the distribution we would get. Here we go. This is the shape of the distribution we would get if there was no difference between red and blue rates of winning. Now the p-value we're going to calculate is going to tell us how likely it would be to get the proportion we got, or a more extreme proportion, on the assumption that the null hypothesis is true. So keep in mind that this is the probability value of the outcome that we obtained if the null is true. What we do here is we take the probabilities of the outcome we got and anything more extreme than that, so that's 16 through 20. And because we're doing a two-sided test and just looking for difference, we're also going to take 4 through 0 at the other end of the scale too. So these two tails are where the 2 here comes from. These are the size of those small columns, and this is the total that those columns add up to. So that's a proportion of the 1 that would be the entire area of this graph we're looking at, and 0.012 is the proportion of the red that you can see shaded in. And that is equivalent to the probability of obtaining these outcomes that we obtained if the null is true. Now what we do next is we compare this to our significance level. We're going to explain significance level much more in detail in lecture 15b. For right now, we can just think of this as the criteria we use for judging the outcome of our test. And it's on the basis of the p-value compared to the significance level that we'll decide to reject or fail to reject our null hypothesis. Essentially, if the p-value is less than or equal to alpha, then the null hypothesis is rejected. And the typical value for alpha is 0.05. It's a flexible thing. We can move that value for alpha if we need to under different experimental conditions, but keeping a ballpark in mind of 0.05 works. Okay, so what do we do now? Well, we have our p-value calculated, 0.012. We compare it to alpha, which has come out as A for some reason in this slide, but this is alpha. And in this case, P is less than alpha, so we can reject the null hypothesis. That means that we believe that HA is true. However, we're going to express this as we reject HO. So P is equal to 0.02, therefore we can reject HO. And in this experiment, we got sufficient evidence to allow us to reject HO. Red and blue shirted athletes are therefore not equally likely to win. You can see that is the HA that we wrote at the start of the experiment, isn't it? However, the terminology we use is that we reject HO. And listen into lecture 5b again to understand why we use that format. So reviewing our data, we can say that the red shirted athletes did win more frequently than the blue shirted athletes. Okay, here's another example. In this example, we're looking at the resemblance between dogs and their owners. This is something people often comment on. Do we pick dogs that look like us? And there's some nice examples in the images here. In this experiment, 41 dog owners were approached in parks and photos taken of the dog and the owner. The photo of the dog of the owner and then along with a second photo of another dog were shown to students to match. There were 41 cases in the database, one corresponding to each pair of dog owners and their dogs, and then that second picture that was introduced. The data set therefore comprised the number of those dog owners and dogs that were correctly matched by the majority of people that took the test. Okay, so if dogs don't resemble their owners, the proportion of correct matches should be 0.5. There should be as many incorrect as correct matches. If dogs do resemble their owners, our HA is dogs resemble their owners, and then the proportion of correct matches would be different to 0.05. Okay, so you can see we've set this up in a very similar way to the way we set up the wrestling bout investigation. Okay. So we had our 41 matched pairs, and of these 23 were correctly matched by study participants and 18 were incorrectly matched. So our sample proportion, 23 divided by 41, is 0.56. So again, we look at the null distribution, the frequency of the results we would have obtained if the data was sampled under the null condition, okay, i.e. there is no resemblance between people and their dog owners. So again, we've got this white distribution, yeah, and then we've shaded in the red portions that would account for the frequency of the results that we obtained. So that's 23 correct and 18 incorrect. If we were then to calculate the actual area of all those red shaded columns, our value would come to 0.56. So again, we've got the proportion of the area under the curve that accounts for the outcome we got and anything more extreme. Convert that to a probability of 0.56. We compare that to our level of significance, 0.05, and in this case our value is not below 0.05, is it? So in this case we don't reject the null hypothesis, and our evidence suggests that there isn't a resemblance between dogs and their owners. We basically have no evidence to support the idea that owners resemble their dogs, or dogs resemble their owners, depending which way you look at it. Okay, so if we put this all together, and maybe leave you with some take-homes to make sure this is all clear and in place. We're creating frequency histograms that show the distribution of the data we would obtain under the conditions of the null hypothesis. We refer to this as the null distribution. In the case of the dice we can see in front of us, that would comprise the frequency of all the different scores on the dice. Yeah. We then take our experimental data, we establish a probability value for the experimental data. The area of each column here is equivalent to the probability of obtaining each score under the conditions described in the null hypothesis. And so what we can do is use these probabilities to determine the probability of getting the value that we got. Imagine we got a score of 12 5 times in a row from a pair of dice. We can see here that that's very, very unlikely. So it would seem that in this condition, the dice that you had were loaded or something. They didn't adhere to the conditions of the null hypothesis. Okay. When a result is so extreme that it supports an alternate hypothesis, that's when we say we've got a significant result. So essentially what we've got to keep in mind is that statistical testing sits firmly on the principle of repeatability. The idea is that we do our experiment, we obtain our results, and we evaluate the likelihood of obtaining these results if the null hypothesis is true. So it's the p-value we get is the probability of obtaining our results under the conditions described in the null. Where the result of an experiment is so extreme that it seems very unlikely that you would get these results under the conditions described in the null hypothesis, that's when we say we have a significant result. And what that's essentially saying is that if you try our experiment, you'll get the same outcome. And we think we've established a repeatable pattern, difference, or trend. Okay, well, hopefully that lays down some of these principles in place for you. Feel free to come to me with questions about this, think this over, try the worksheets in the books, and we will be discussing this again in class. Okay, pop over now to CI Learn and you can try quiz 5A, and I shall see you in class. Thank you.